we will have the Q&A session with our panelists. I would like to invite all the 7 speakers to the podium, and also the 3 speakers from the session 1 this morning will also join us as panelists as well. Dr. Xiaoming Xu, Dr. Jeff Boston, and Dr. Willis Smith.
In order to
we have two microphones in the room. So for in-person attendees, if you have any questions, please use the microphone. Yeah, for the question.
Thank you all. So this that hasn't done as hands-on, so, but I've done quite a bit of rheological characterization. So for something that's a non-Newtonian solution, what about the idea of predicting or calculating the 0 shear viscosity at a given temperature, and then using that for your DLS measurement? Has that been tried, and what drawbacks are that approach? Thank you.
Yeah, so I brought viscosity up last, so I'll go first. So I do like the 0 shear viscosity approach. The 2 methods I'm familiar with for that, the simplest one is simply adding beads of a known size to your dispersant matrix. That works great if you have your dispersant matrix. It's not going to work when you have your large particles floating around, so you can't really resolve your beads, your bead standards of known size. The other approach is to just take a viscometer that continues to, we'll say, go slower and slower at less and less shear until you reach a plateau and then extrapolate. That was something we didn't reach a plateau with our instrument in this case, but those are the 2 that I'm familiar with. I think they're a valid approach for evaluating your dispersant in this situation. And then if you can't, then you work with the next best tool that you have, which is higher shear or dilute till you can measure.
Well, I like this on. Is this on? Okay. Yeah. One more question I would ask is, how did you get to a non-Newtonian liquid? And if it's coming from a very high particle concentration or a high polymer concentration, then that can affect your measurement significantly. And so there's probably a first question of, why are we in this regime?
Jonathan, you have any insight?
Yeah. So one of the older versions. Do I have to press it? Testing. Okay. But one of the versions of the Zetasizer had a microrheology capability where you could use tracer particles, as Kevin mentioned, to calculate viscosity. One of the requirements is that the tracer particles cannot interact with the surrounding dispersant. Otherwise, the size would change, and then that affects the accuracy. You're basically using, it's like a reverse application of the Stokes-Einstein. So you absolutely need to have tracer particles that don't change size in the dispersant. That's very important.
Yeah, and then what I would perhaps add is that there is a really important distinction between suspension viscosity and just the viscosity of the effectively the placebo, so the formulation without the particles. I think in general use you would want to use the second one, and then there is also what's maybe an important consideration is the effect of your excipients that would normally contribute to viscosity. But if you add particles, they would actually adhere to the particles, so they would not contribute to your viscosity. I think that's more for the rheology approaches, more of a relevant thing. But the difference between those 2, depending on particle concentration, can be quite big.
Just want to see anyone has additional insight for discussion.
So I brought up microbiology as well, and one of the things that is important with microbiology is understanding, as Jonathan said, that the particles interact with the sample. And I'm sorry, I've completely lost my train of thought now. I'll come back to it. I'm sorry.
Do you have a question? Maybe we can, yeah.
So from my perspective, particle size is important not only for generics, but also non-generics. It's applies to PLS and I guess a lot of things are diffraction. So the question that I have is with regards to validation versus specification setting, right? So typically you validate your method and then the specification setting comes afterwards, right? The question that I have is, if my specification is very wide, say as an example, E50 to, I'll use laser refraction as an example, E50 to 10 to 100 microns, that's my specification range. Does my validated method have had to be validated on that size as well? The reason why I ask this is, sure, my specification range is 10 to 100 microns, but my clinical material, my commercial material is often a very tight range within that, say 20 to 30 microns. And we're all taught to validate our methods on representative materials, right? Which would be within those 20 to 30 microns. So,
First, I do echo your point that although this workshop we're kind of focusing on the generic product, but particle size measurement is a very universal for both generics and innovative kind of brand name product as well. So on the point of setting specification, I would say one of the important thing about why we set specification is to ensure that every batch that we produce can produce exact same or behave the same way, especially for an innovator company product, right? Is to ensure that the batch after batch after batch, they all perform, behave the same way as those first entering into clinical trials. Or in the other way, we set a specification not because we could, setting a range that we can manufacture them, but rather we set the range such that we know the product that we manufacture batch after batch, they do the same function in vivo, in human. Therefore, I think it's not just about the range that, 10 to 100 micron, whether or not the measurement can adequately be shown to be suitable for its intended purpose. I think even more important is whether or not that range clinically is relevant for its intended purpose. And the measurement range comes second. The purpose of that setting that specification come first. So with the range that we know what is clinically relevant or important, or we're setting a size range because we consider size being a critical quality attributes, that we know that changing the size by 10 micron or 100 micron can in fact impact the dissolution or impact the bioavailability. Therefore, we might need to set a tighter range for acceptance. So that I think should be the main focus or purpose of setting the specification, which is not based on what the technique can provide to us. But on that argument, I think completely change when you are developing a generic product. Because when you're developing a generic product, your comparison is no longer just clinical data or how we said what's useful or effective or not effective, but rather is to compare with the product that's already on the market, which you do not need to do a clinical study most of the time. Therefore, you want to use a measurement to prove that it's comparable. now becomes challenging. If the innovative company product has a specification that's very wide, now the comparison between generic and innovative company product become meaningless because by ensuring the value is comparable has no bearing on whether or not actually the product perform the same way it should be in vivo, therefore by equivalence, right? So therefore, in this case, if the innovative company product setting specification is too wide, it may actually hurt or make it very difficult to compare a generic product, which is one of the common challenges we see, is it how do we set the right range of specification? Hopefully I'm diving too deeper into that range, but thank you for the question. And thank you for being here. I know you're coming from a innovative company, so.
Just want to see any other panelists have any insight for this question? Yeah, maybe we can move on to that.
I know the answer to almost every part size question is, it depends. Poly dispersion can be an index, can be an issue with DLS. How do you determine when you have a problem and you need to do something?
Johnson.
So I would look at your other. Testing, okay. I would look at your other data quality indicators. So I can speak to the Zetasizer, and it's in regard to, like, can you trust this PDI result? Or is it interfering with the measurement? So I would look at your other data quality indicators, which would be like the y-intercept of the correlation function, the attenuator that it's being measured on, and maybe the derived mean count rate. And if all three of those look good, then you probably have a true valid PDI that's reliable.
So another value to look at is your polydispersity index. So years and years ago, Ben Chu claimed that polydispersity index values over about 0.3 is where cumulus analysis starts to fail. And I certainly agree with those other measures, but your polydispersity index is a good clue. A second good clue beyond what was already mentioned is your orthogonal sizing techniques. So as you go to laser diffraction or microscopy, and you kind of get those, hey, that's funny questions, that's where you start to really worry.
I think it's also worth looking at orthogonal techniques such as introducing chromatography to the system in flow mode and using that to separate out the very broad distribution that you're talking about, to give you more information. So yeah, additional techniques that utilize the DLS system might help you understand and characterize that system.
And I think one thing I would add, I think I heard it down there stated pretty well. Looking at your data overall can give you some good insights. If we're thinking about the transformation that we use to get to polydispersity out of that autocorrelation function, it's the second term in an expansion. So if you're looking at low signal to noise, you may still be able to get good data out of the radius measurement, but you start to see more noise out of the polydispersity. It's the second term, so you see that happen first. So looking at your concentration, looking at your signal to noise, is the system dirty? Do you need to increase the laser power, decrease attenuation? Some of those things can play in there as well. Just thinking about the overall picture of how we're getting to that data.
Xiaoming.
I think one of your underlying of your question, there's an assumption that polydispersity or seeing the sample have or exhibit polydispersity is something we need to avoid, which I think it may not be necessarily the case. As I indicated in the opening, that we don't have an ideal material to begin with. What's in the drug product is what's in the drug product. Whether or not it exhibit a polydispersity, we need to look at it, having ability to characterize it to the best possible ability that we can tell what is in there. if that means we need to reach the conclusion that the sample is polydispersed, but if we have confidence in making that statement that how we derive or get into that conclusion about polydispersity, and also keeping in mind size is only one measure of the product. As we prepare the product getting to patient, we have other means to ensure we have set adequate control to ensure that the product behave as it should be. Therefore, we don't necessarily need to avoid or have to pretend that we only have multi-dispersed particle populations. Again, I'm stating something that may be true, but not necessarily, I guess, wide accepted, that we don't have always the most ideal sample to measure. Therefore, it's always going to be challenging.
Yeah, just to kind of piggyback on this, at NCL, we get a lot of samples, we characterize different attributes here and there. At the end of the day, size, polydispersity may not matter, right? Your specifications have to be linked to a biological outcome, good or bad, right? So it might be the case where size may not be a critical quality attribute in this case. You have to look at maybe drug loading, drug release, things like that. So just keep that in mind.
Yeah, I think, hold please. Hold for microphone adjustment. Yeah, I think kind of to the point of, is it a feature or a bug in the system that you're looking at, as kind of Xiaoming said, is, polydispersity just exists. So it's really goes back to what we were talking about with method validation, is like, are you certain that the polydispersity is, reproducible? If you can get it the same every single time and it's not a variation between measurements or between some methodological sample preparation procedure or something, then it really exists and it's a feature of your product. And so that's kind of, in my mind, the key is that you look at it and you don't go like, oh, actually it's just a variation between measurements. Then that is more of a question of your method optimization process. So those are kind of the things that you have to think about when you all of a sudden see like, oh, my polydispersity is going up when I dilute it or when I do this. You have to kind of like make that justification as to what specifically in the process am I doing that could introduce some variability in my measurement. Because in the end, it's about being certain about your measurement. So I guess I'll just end there.
It's going to say, like, fusing together Xiaoming and Williams' points, like, without context, data just exists.
Maybe we can, next question.
I have a two perspective. First question is to Jeffrey. So in your presentation, I think you showed one of the intensity-based distribution where I think multimodal distribution exists, and you showed that after filtration, monomodal distribution. So when and where, like what products we can do this kind of filtration? I can see clearly some multiple populations, and these are really representative of the native sample, and like how we will approach this filtration.
This one. Okay, sorry. So, sorry, repeat again. Sorry, with the intensity versus volume and number, you're saying?
Intensity distribution, which were multimodal distribution.
Yes, yes. Oh, with the Dendrimer example, right? Yeah, so in that one, we're using, well, the example I showed, there was intensity, right? They had multiple peaks in there.
Yeah.
And then we use volume distribution for relative amounts.
The same time, for after filtration.
Okay, yeah.
When we can do the filtration?
Okay, when? Okay, yeah, that's a good question. So when I said earlier, filtration, typically I don't filter, right? I typically take the sample as is because I want to know what's in the vial. So unless SOP calls for filtering through whatever micron filter, then I would filter to run the sample and try to compare our results with our client's report. Now, if you're just looking strictly at, let's say, metrology point of view, and you have a polydiverse, messy sample, and in this case, my particle of interest was small, right? Less than 10 nanometers. We know with scattering, it's functions to the 6th power. Anything larger will skew, will basically dwarf out that smaller population. I won't see it unless it's in appreciable amounts. So then I will filter the sample if I know ahead of time that my sample should be, let's say, less than 10 nanometers. Does that make sense?
Yeah.
Okay.
Can I add something real quick? if you're like doing an intermediate quality control thing inside of a process where after that process you filter your product, by all means filter it before you analyze it, right? But that's like, if you're not filtering the end product, right, don't necessarily come at, you know, a method by being like, oh, you have to filter before you measure this product. Is the filtered product being delivered to the patient, or is an unfiltered product being delivered to the patient? So just to kind of hop in there.
Yeah, and I'll caveat that as well. When you filter, you might also, you know, extract out other components in your system. So just keep that in mind as well. You can pull out API, you can pull out excipients, components of the formulation, which will make it unstable. So keep that in mind when you filter.
Thank you. So my second question. Kevin, when you showed the iron sucrose data, which dispenser it is being used, like?
I'm sorry, can you repeat which data?
iron distribution data what you showed.
Oh, the distribution data I showed was generally mass distribution.
Dilution also, like whether it is done with water or sodium chloride.
Oh, I think it depends on the drug that I was talking about. In one case, I think, yeah, go ahead.
Like when you showed the data, it's 10 nanometers. Like all the reported journals everywhere, iron sucrose usually 10 nanometers, the zero. But I think when from what is data, sorry. So when you showed that it is a 5.6 nanometers. So which is actually iron core side, which is not the iron which is sucrose. So how did you get that core side with the DLS? That's what my question. I just want to couple with that. That's
Yeah. So really good point to clarify. The data we showed was hydrodynamic radius. So measured radius of 5 to 6 nanometers, so measured diameter of 10 to 12 nanometers. So it's an important distinction to make, that sometimes we need to be precise with the label that we give to the data. Historically, some people think about size in terms of radius, usually from more of a physics background, but I think most folks in industry and in this audience probably think of things in terms of diameter. So it can create confusion. So it's a very good point. I probably should have labeled that better as diameter and not as radius. And so appreciate that.
initially I thought it's an iron core you are measuring. So it's not like an iron core we are measuring with the DLS, right?
Correct.
I've got a very basic question for Dr. Claus. Here, like she talked about intensity, volume, you mentioned number, but what information is pulled out of that aspect?
So what do you need to do that transformation? Are you?
What information is pulled out? Talk about like intensity, size.
Yeah. Yeah, we typically don't use number. I know people do use numbers sometimes for comparison. Like if you know exactly what you have, kind of like what an example I showed with Z average with two different QCs for batch to batch consistency. I've seen people use number for that purposes, where they just under the same exact conditions, same everything, they use number to compare quickly, but they don't use that to report any sizing. So then I can see people using that, but typically we try to avoid number altogether.
Yeah, so I have a comment on the distributions that you use. So I think of them as two ways. One is translating data to a different language, and the other one is as like actually a computational data filter. So here's the analogy. When you're driving in your car, do you look at the RPMs of your tires? No, you look at the speed you're going. So looking at intensity distribution may not necessarily mean anything to you, but looking at the distribution of your sizes relative to your mass may mean a lot more. If you're dealing in the world of LNPs, looking at that by mass maybe doesn't make a lot of sense, but maybe making in terms of numbers makes more sense. So changing, it's more about the language that you're picking is one. And the other thing is that it's skewed based on size. So they're basically each a low pass data filter that's going to skew your distribution further and further to smaller sizes. So that was actually interesting where we had one slide, where we were looking at, in Jeffrey's slide deck, where we were looking at, here's a physical filter, and right next to it was a computational data filter, where we were looking at intensity distribution versus number distribution. So just be aware that as you change the language that you're using, that you are also filtering your data set.
So
for intensity-based distribution and number-based distribution, if sometimes you are in, it depends on your interest actually also. So if you have like monomodal distribution, you'll see in most of the cases all are same, like number, volume, intensity. But whenever you are like looking at if the progress of the reactions or agglomerations, you will see the difference in the number-based distribution, like this number is actually decreasing, another peak is increasing or not. So that will help you to see the progress of a reaction when you are focusing on the number-based distributions. But in the DLS, we mainly focused on the intensity base, yes.
Next question.
So when we recommend comparative characterization studies, depending on the characteristic, sometimes we will recommend orthogonal approach, right? So here for particle size measurement, historically, I think DLS and MALDI has its own character, but now it's a new technology, the size range, it can cover so much large. So we always have this curiosity question, right? When say drug product for components purpose for comparison, is there a possibility for one measurement we say is equivalent, but it's not using the other? So when do you think it is beneficial to potentially look at LT versus DLS? And do you think the measurement comparison without the always agree with each other?
Who want to take this question?
Well, I guess I'll start. So, so first I'm going to ignore the question about orthogonal techniques for the moment. So we'll just set that aside. So as you're really measuring two different things when you comparing dynamic light scattering versus laser diffraction. And so you're going to expect in DLS, you'll have a hydrodynamic size. You'll typically take a Z average. And that comes out of kind of weighting uncertainty of data. Laser diffraction, you're going to report the size of a sphere that scatters the same way as your particle. And so I wouldn't expect both methods to match for any practical sample. And you're going to gain different insights into your sample depending on which technique you use. If you're early enough in development, you could pick what you think is most important to work on. Or you might find additional insight from a second method, just as reference data or something like that. But you're really asking two different questions of the sample as you switch methods. We just happen to call them both particle size.
And I will also want to mention that, so dynamic light scattering is mainly depends on the Brownian motions. And once the particles get bigger, the Brownian motions is not that much effective anymore. So that's why like at that time you have to depend on the laser diffraction. So if the particles are very small, at that time Brownian motion is much more effective. And their DLS actually works very well.
Yeah, I'm just to clarify one thing, where even for the same product and the different companies using DLS, for instance, we don't expect the data to match. So when I say the result of is, for instance, if we conclude those are equivalent using DLS measurement, well, we have the same conclusion. We're looking at data just LB on its own, not to compare the two. The first principle for each technique is for the difference we described. So that's our curiosity. I think that's probably means another level is when you apply statistics. Then the inherent variability associated with certain measurements would play a role in terms of that kind of assessment. So that's where it's not very clear, but I feel like that's more discussion as well for bioequivalence that is when you compare side by side reference to generic, then that's a fair comparison. And then we are looking at the conclusions whether they are equivalent. So we are always curious if one says this is equivalent, the other says not. But we haven't seen a case, but that's just a curiosity question.
So what type of size didn't match with your like both of the results? Is it?
Okay. This is very clear, it's upper micron than see around just the 5 micron or upper range. then you can start to see historically there are people discussing RT is not suitable to measure nano-sized particles, but now it's actually extended much below to the conventional size range because of the principle. So that's where we start to see then in our guidance. If it's very conventional and clear, we see the average and PDI, but then some cases we see 10, 50 and STEM. But then sometimes we do see people use, for instance, DLS and then they convert, which we would prefer. But then again, if everything seems to be correct, then we still, because it's comparison purpose, right? We not necessarily reject the method if it's probably validated.
So like when it's from my personal experience, I saw that if the particles are suppose 1-2 micron, that's not a good for the TLS, because the tail on the like higher end particle size may be above the range of the DLS. So that's why you will see variations, maybe smaller particle size in DLS, but a larger particle size in the LD. But it happens exactly opposite when it is in the smaller particle size range. So that's what happened, because at that time the LD cannot capture the particle size, which is very smaller in particle size range. So that's what you will see. But if it is in the like a couple of 100 nanometer to micron size range, yes, both can perform very well actually.
I'd also like to add just a little bit of information. It is possible to introduce an orthogonal technique such as sedimentation, which is able to extend the range of the top end of DLS and expands over to the laser diffraction measurements. So there is orthogonal techniques that have been developed to cross that border between those two techniques to give you a better comparison.
Thank you. Maybe we can, yeah, next question.
Okay, so today we saw elephant 2 times, but we also addressed the third one. So what is about specificity and selectivity of the method? So we know that due to the nature of the method, directly possible, but my question goes to the vendors, what are the current advances? Where are we here? Let's say in some cases we are comparing compound generics. When we are comparing final products, this means that these compound generics, usually they are not clear, homogeneous, uniform, single particles, but they are in a complex matrix where we will take some steps. Where are we here?
I guess I'll start. If you've got a complex system, DLS is really not an appropriate technique for measuring that sample. You need some kind of separation method to be able to identify the different components within your system. So I would argue that perhaps DLS is or DLS plus something like a separation technique like chromatography might be necessary.
And is something happening here? Like currently, let's say we are DLS, we have a formulation, nano-size based, where we need to measure particles. What can we do?
So if you look at DLS, you're really looking at how the particle moves. So if you have a complex matrix that affects particle motion, which kind of ties back to my comment about non-Newtonian liquids, you're really not able to make the kind of claims about particle size that you can make about a more simple system. And if you go really complicated and you have a complex system where the particles are stapled down or inside other larger particles, kind of hope is lost. And now you're looking at microscopy or some other technique like that. Sorry.
But in case you have, let's say, not such a complex, but maybe several different particles which we can measure. Let's say the complex is clear, but maybe we have two components.
Okay, so now we're moving in a direction where, well, so here I would start to look at something like nanoparticle tracking analysis, where, and you know, I'm sure Wyatt's can mention field flow fractionation, where this also applies, where you start to separate the particles. And if you have 3 different sizes in your formulation, you can separate them and either in the measurement or in the fractionation and measure each of them to differentiate. Another technique would be to tag your relevant particles and do nanoparticle tracking, which will let you just look at your particle motion for those particles. I'd go back and worry about viscosity, but you might be able to do something about it because now you're just looking at your analyte of interest and not the rest of the mess. It'll be exciting.
Yeah, Jonathan.
Let me add, keep in mind that DLS is an ensemble measurement where that scatter volume, you're measuring tens of thousands of particles or more, and you generate a single light scatter trace. That's the light scatter coming from all the different populations of particles. So of course you can separate them or resolve up to 3 to 4 different size peaks, but you're not really characterizing what material that those particles are made out of. So unless you couple DLS with something else like chromatography or another purification process, you're not really going to get extra specificity using just DLS.
Just to add a further comment, there is a technique called resonant mass measurement, which uses the buoyant mass of the sample, which would be able to identify differences between things like liposomes and proteinaceous materials. But they're hard to get hold of now because they're no longer manufactured.
Thank you. For next question.
So I was having this conversation with Camilla a little bit, but I wanted to extend it because one of the things I didn't see in the presentation when you do dynamic scattering, also in diffraction applies as well, is you often see a artifact coarse peak that show up when you do that. Anybody who has run iron sucrose on dynamic light scattering would have seen it at least once, right? In my experience as well. So the question is how do we deal with this? Because when this shows up in my validation, so if I do dynamic light scattering, it'll throw off my PDI, right? If I do dynamic laser diffraction, it throws off my D90. So should we take an approach of not validating those metrics so that it's fail, because it's not because of my product, it's coming from the instrument itself, and I know that it's an artifact, or is it better to treat it away? So to demonstrate, have technical justification for removing that as a result of an instrument issue, and then using things like fine particle mode. But then if I do that, how do I validate that whole thing? Because now I need to demonstrate that I won't see particles, say, a millimeter in size. So how do we, what should be our approach there for DLS, laser diffraction?
So I think it might be good to go back to the root cause of it. So I'm not fully in agreement with that. It's an artifact. I think what you're seeing in that product is actually just particles from the sucrose. So it's a contamination, and then the artifact aspect of that is maybe more related to number fluctuations due to the fact that there's only very few of those contaminants in your sample.
So disagree with you there in that you don't just see this on iron sucrose. You also see this when you don't use iron sucrose. So for instance, I don't iron sucrose anymore. We also know that when you go from one vendor to another, same dispersant, it goes away.
Yeah, that can be correct. So I think that depends a bit more on, for instance, how you analyze the ACF in a particular sample, how much specificity you can get for those kinds of analytes, and how much of the correlation function you actually use. So, for instance, one of the things we found with this iron sucrose is that where we would typically use only 95% of the autocorrelation function, for instance. If you then basically stretch that to include 98% of the autocorrelation function, this artifact is resolved and becomes effectively a particle contribution.
So that's essentially what you're describing is a form of digital filtering.
Correct. Yeah.
Which, if you use fine particle mode laser diffraction, it turns off the initial detectors. In essence, is a digital filter. But from a product quality perspective, we obviously want to ensure that there are not big particles that we're not accounting for because of, say, a manufacturing issue that resulted in metals in my system, right? So from a validation perspective, even if I have technical justification to use some sort of digital filter, I would think that we still need to demonstrate that we're looking at it from a holistic perspective to ensure product quality, right? So, which then needs to feed back into my validation approach in the first place to account for it, you know? So then, what approach do we take?
That's a very good point. So I can give you our perspective on the situation, which is that we open up our model. We have full customizability of all of these digital processing parameters. So fit limit I described, you can give a value between effectively 0.1% and 90%. I would not recommend the latter one. But simply by giving the end user more freedom there, we try to kind of circumvent that. There's other approaches. I think there is even ISO recommended values for these types of, for instance, for the fit limit and so. And we didn't even touch on the smoothing factors. But that's kind of my perspective on it.
If I can add some context to this, I think if you're talking about a peak that turns up in the micron size range within a DLS system, that's often caused by a misfit of your correlation function. So you need to look at the data that you have. The misfit occurs at the bottom end of the correlation function due to noise in the system. So some of those artifacts that you see are actually not real, and it's caused by the mathematics. So you need to make sure that the correlation function in your data looks like it should. If the fit is failing at that end, then as was mentioned, you may need to filter those data points out and isolate yourself to the top end of the correlation function only. So I think there's a little more complexity to the question that you have. Is it real, or is it just an artifact of a poor fit?
You filter the sample. And it's the obvious thing, filter the sample and see if it's there. And if it's there on the filtered sample, you know it must be an artifact.
Yep, exactly. Yeah, we've done that in the past as well to exactly demonstrate that it's an instrument artifact. It's not our sample.
This feels like a great small group discussion question. So sorry.
Okay. Thank you. Thank you. Sorry about that.
Yeah, we're a little bit over time, maybe. Yeah.
I have one quick question. If the RLD reference product is highly variable from lot to lot, how we can proceed in finalizing the final formulation and specification? And also one more question, in case only one batch of RLD available in the market, how we are going to finalize again?
That's a great question. That's a question we know generic industry struggles a lot. And then I wouldn't say we have the answer for it. And I will call you, this afternoon we have a group discussion. So let's have a conversation on that topic.
Thank you.
Actually, we have a question dedicated to that. So let's have more discussion among all of the audience here today. But that's a great question.
Thank you, sir.
Yeah, just for online applicants' benefit, back to your question, RLD variability is more towards once you have a validated method, your measurements and your data comparison if you only have one lot. But in terms of method validation, RLD is certainly suitable and maybe even desired. But validation-wise, you can also use your own test product. So it's not only to the RLD, just so that's just a general statement for method validation samples.
Yeah, we are. Thank you for the discussion. We're a little bit over time. Maybe we can take 1 more minute from online questions. After that, we will conclude the discussion.
Okay. Yeah, so online we have close to 1,000 attendees and they have submitted multiple questions. So to save time, we probably only pick up 2 of them and 1. Okay. Okay, so I think this is more like, I believe it's more beneficial for those like generic companies if we just give this question to like agency. So the question is, regardless the technique and the instrument, it seems like the complex generic sameness to the RD and the generic formulation is all that matters. So perhaps the technique can be addressed as part of the OGD's PSG. And perhaps instrument and conditions can be also be recommended. So I think this probably a lot of people, they just want to know, yeah, whether like a PSG can cover more about like techniques and instruments, which can sometimes reduce, they have some selection like levers, yeah.
I guess I'll start. So for a disclaimer, I do not write PSGs. So I work in the laboratory. But often for like complex generics, we do a lot of internal research, and a lot of times we do publish manuscripts on specific products using suites of different analytical techniques. And it might not just be like one, and we're not saying like, hey, because we published this, you need to do it this way. There are a million ways to do it. I am all for specificity. So I don't know if going forward, we're going to start including like some references or some more specificity in PSGs. But I love that. And I think if you kind of keep your eyes peeled, you know, you'll see a publication or two out there, maybe on a product that you are specifically in. And by no means can you not do something different. So if you have one instrument and we used, you know, or in the PSG, we say DLS, and you want to use laser diffraction and it's appropriate, do it. But I think, yeah, in general, I don't think we will ever stand behind necessarily being like, you have to use Unchained Labs, do this exact thing. You know, that's not our, as much as we might want to, that's not our mission, is not to back any one specific vendor or instrumentation. It's about making sure that you are reporting to us the data correctly thoroughly enough that we can be certain in your measurement. Let's say that.
100% agree with Billy have said. Just also add that PSGs reflects the current understanding that the agency has on a particular topic and the recommendation that are made based on what's current understanding. Therefore, we have to strike a balance between being overly prescriptive versus still offer opportunities for the generic companies to innovate. When we have understand enough about a product that we can make certain recommendation about the measurement, you might see some of the PSG has very specific recommendation, either laser diffraction or DLS. And again, those will be based on what's best understood already about the product. If it can be made, then will be made. But otherwise, as Billy said, you know, even if we made a recommendation on DLS, but you develop product, the measurement based on laser diffraction, that can be adequately justified, that is equally, acceptable. But then become, how do you, to show, as we have shown this morning, all the what should be reported, what's the context, all the associated validations to show that you have enough understanding about your measurement, you have enough confidence and trust about your results, show that to the reviewers so that they can then assess, determine whether or not the submitted data adequately support the conclusion. Again, I understand the desire for more specific, more prescribed detail in the guidance, but sometimes we hear the other side of the argument too. Don't limit us what we can measure, give us some flexibility. So, strike a balance. That's sometimes you cannot make everyone happy, right?
Yeah, and I just want to also add that the people who are writing the PSG are doing a great job. And as Xiaoming has said, we are always learning new things, and the PSGs are not set in stone. They are updated. So when new things are learned, when a better way to do something is, developed, that's usually in the draft pipeline already. So I just wanted to add that out. So they're not set in stone. Keep your eyes peeled.
And lastly, in generic industry, you're paying the user fees for that. You are supporting the GDUFA research, which allowed us to, better understand the product and better make better recommendations. So that's, all because of the industry has been supporting it.
Thank you for the great discussion. I think that concludes our panel discussion today. Please join me to thank our panelists and speakers. Thank you. So just a couple of announcements for the virtual attendees. I think for this session, we'll conclude our virtual session today. But just like Xiaoming mentioned that CRCG already uploads complimentary videos and materials in the websites. If you are interested, you can further look at those. Those are very useful information as well. And the other reminder is that tomorrow we will start the workshop at 8:30. Please join us on time. And the other thing is that we welcome online audience to further submit questions. If your question hasn't been addressed, we will try to see if there's any other way to address the questions. We will include that in a discussion in our afternoon in-person discussion as well. And also, all the recording and the slides of the sessions will be posted about like 2-3 weeks later. I think CRCG will send you a notification once it's posted. Yeah. Thank you, everyone. Thank you.